Okay, so this misconception video is all about DNA structure and extraction. So the first thing, this is from B3. The DNA extraction practical is the core practical in B3. So, starting with the basics, DNA was discovered by Watson and Crick. Well, it wasn't discovered, they discovered the structure of DNA. That is horrendously drawn. This one's a bit better. There's your double helix structure. And we say that it's got a sugar phosphate backbone. So DNA is two strands wound around each other to get this double helix. Now, the sugar phosphate backbone, you've obviously got two for the two strands. There's one here and there's one here. And you'll see what I mean by the sugar phosphate backbone in a moment. You've then got the bases in the middle, which are complementary to each other. And hopefully you know that there's 4 potential bases in DNA. Adenine, which is obviously shortened to A, always pairs with thymine. And then you've got guanine, which is complementary to cytosine. So they're given A plus T, G plus C. And one way of remembering it is just the way I remember, at the garden centre. And these bases are complementary to each other. So for example, if A looks like that, C would look like that. Okay? They are fully complementary, they would fit together. They actually join by hydrogen bonding. So if this is my molecule of DNA, I'll draw a different one because that one's going to be too hard to label. The bases match up along the middle, and then there's your sugar phosphate backbones. Okay, but DNA is actually what we call a polymer. A polymer is something that's made up of many repeating units. So what is the repeating unit that DNA is actually made of? Well, we say they are made up of nucleotides. And you need to know what the structure of a nucleotide looks like. So a nucleotide, the general structure of one looks like this, where you have the phosphate group joined to a sugar group joined to a base. And that's one nucleotide. And remember, that base can either be A, T, G, or C. It can be any of those 4 bases. Now, what happens is that's just one nucleotide, and obviously DNA is made up of many of these. These connect to more. There's your phosphate, there's your sugar, there's your base. That then connects to another one. That then connects to another one. And so on. And then this is why we say it has a phosphate sugar backbone, because it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, with the bases connected in the middle. So there's your sugar phosphate backbone, and then your bases. You'd obviously have another strand here, which would be complementary to those bases. So one of those, just one on its own, is a nucleotide, which join up to form your polymer, which is DNA. Right, extraction. As I said earlier, this is the core practical. So you would have done this core practical with kiwi or strawberries. You can do it with peas. I think that's the example they use in the textbook. You could do it with carrots, anything, but fruit tends to work the best, like strawberries. The first thing you would have noticed is your teacher had probably blended the fruit. And that's just to break down the cell walls. Because what you're trying to do is you're trying to get the DNA out of the nucleus. You're extracting it out of the cell. So you blend the fruit, that breaks down the cell walls. The next thing you do is you would have added a detergent. And this probably would have been like a washing up liquid in the lesson. So you add a detergent. What that does is it breaks down the cell membranes. So it breaks down the cell membranes. Now that's useful because if you break down the cell membrane, you can get inside the cell. It also breaks down the nuclear membrane. So not only have we broken the cell walls to get inside, we've then broken down the cell membrane to get inside the cell. We've broken down the nucleus to get inside the nucleus, which is brilliant because that's where the DNA is kept, is inside the nucleus. What you then would have added as well is salt. And salt just helps clump the DNA, just to make it easier to extract and for us to see. You then would have added protease, and it wouldn't have looked like protease in your lesson. It would have looked like eye lens solution for contact lenses. Now, what the protease does is it breaks down proteins that the DNA is often wound, well, the DNA is wound around these proteins. So if you use protease and break down those proteins, it frees the DNA up, easier for us to extract. The final step is you would have poured in ice cold ethanol. And what that does is it precipitates the DNA. That means it separates it from the rest of the solution. So if this is your test tube, this would be your solution, your DNA would go separate to the top, okay? So you'd end up with your precipitate of DNA. And it looks like white strings. And you actually should have been able to, in the lesson, to actually wrap it around a glass rod. And that is how you extract DNA.